Intervertebral discs are remarkable structures, flexible yet strong, built to absorb shock and stabilize the spine through a lifetime of movement. But their biology, finely tuned for mechanical endurance, also makes them notoriously resistant to healing. Once damaged, these discs regenerate slowly, if at all, and conventional treatments often fall short of restoring true function. This regenerative failure has puzzled clinicians and researchers for decades, especially given how common disc degeneration is with aging. However, recent insights into the role of cellular senescence may be shifting the narrative. By targeting the root causes of chronic disc dysfunction, namely the accumulation of senescent cells and the pro-inflammatory environment they create, senolytic therapies may offer an entirely new way to promote healing in one of the body's most stubborn tissues. To understand why intervertebral discs heal so poorly, it helps to start with their structure and physiology. Each disc is composed of two main parts: the nucleus pulposus, a gel-like core rich in proteoglycans and water that resists compression, and the annulus fibrosus, a ring of tough collagen fibers that provide tensile strength. What's striking about this design is that it is avascular and aneural in its healthy state. The center of the disc lacks blood vessels and nerves, relying entirely on passive diffusion from nearby capillaries to obtain nutrients and remove waste. This is efficient for mechanical function and protection but presents a major obstacle when injury or degeneration occurs. Without a blood supply, discs can't easily recruit immune cells or regenerative factors after damage. Inflammatory signals linger longer, and anabolic signals struggle to reach the site. This low-turnover environment also means that cellular waste, including oxidative byproducts and misfolded proteins, accumulates over time. Disc cells are often exposed to hypoxia, nutrient deprivation, and mechanical stress, all of which can push them into a senescent state. Once senescent, these cells stop dividing but do not die. Instead, they begin secreting a noxious cocktail of inflammatory cytokines, proteases, and reactive oxygen species, collectively known as the senescence-associated secretory phenotype, SASP. The SASP further disrupts the disc microenvironment. It breaks down the extracellular matrix, inhibits the activity of healthy neighboring cells, and promotes the ingrowth of blood vessels and nerve fibers into the disc, one of the hallmarks of painful degeneration. Over time, this turns what was once a sealed, silent structure into a source of chronic inflammation and discomfort. Traditional interventions like steroid injections or physical therapy may relieve symptoms temporarily, but they do little to remove the deeper biological cause. Surgery can stabilize the spine, but often at the cost of mobility, and does not regenerate the disc itself. This is where senolytics represent a paradigm shift. Rather than trying to override the disc's limited healing capacity, they aim to reboot the system at the cellular level by clearing senescent cells and reducing the SASP load. In doing so, senolytics may help restore a more youthful, regenerative microenvironment, one in which healthy disc cells can once again maintain the extracellular matrix and inflammation no longer dominates the tissue landscape. Early preclinical studies support this possibility. In rodent models of disc degeneration, senolytic agents like physidin, dasatinib, and quercetin have been shown to selectively remove senescent disc cells, reduce inflammatory markers, and partially restore disc height and mechanical integrity. Importantly, these benefits often persist long after treatment, suggesting that once the senescent cells are cleared, the tissue can begin a more normal repair cycle. Some senolytics also show antifibrotic and antioxidant effects, which may further support matrix restoration in the avascular disc environment. However, the same properties that make discs hard to heal, especially their poor vascularization, also make drug delivery a major challenge. Systemic administration of senolytics may have limited penetration into the disc center, particularly if the agent is not highly bioavailable or lipid-soluble. This has led to interest in targeted delivery strategies, such as injectable hydrogels that release senolytics locally, nanoparticle-based delivery systems that can home in on senescent cells, or gene therapy vectors that activate cell clearance mechanisms in situ. These approaches could allow high local efficacy with minimal systemic side effects, a crucial consideration when targeting sensitive or immobile tissues. Another avenue is the use of dual-acting compounds that combine senolytic effects with anti-inflammatory or pro-anabolic properties. For example, A molecule that clears senescent cells while simultaneously stimulating extracellular matrix production or enhancing autophagy could offer a more holistic restoration of disc health. Coupled with lifestyle interventions like improved spinal loading mechanics, anti-inflammatory diets, and resistance exercise, these therapies may help maintain the benefits over time. What makes the senolytic approach so compelling is that it aligns with the natural biology of the disc. Rather than trying to force repair in a tissue that resists it, senolytics work by clearing the very cells that suppress regeneration and drive decay. In doing so, they don't just treat the symptoms of degeneration, they create the conditions in which healing becomes possible again. While human trials in this area are still in early stages, the potential is enormous. If senolytics can be safely delivered into or near the disc in a way that clears harmful cells without damaging healthy ones, we may finally be able to intervene before spinal degeneration becomes irreversible. And that would mark a significant leap, not just in treating back pain, but in transforming how we think about aging tissues across the body.